Evening. My name is Martin Conway. I'm one of the history tutors at Balliol College, and it's my pleasure this evening to invite you to the latest, welcome you this evening to the latest in the series of Balliol online lectures. These have been running for about 5-6 years now. They started in lockdown, and they're one of the good things to come out of lockdown because they're a stimulus to us to communicate the findings of people's research and their general work to wide audiences. And the intention is that the lectures should be approachable, interesting, stimulating ones that provide a sense of the collective intellectual life of the college, which otherwise can be hidden behind walls. So it's very good to have you with us this evening. Our lecturer this evening is Seb Shimeld, who has been the biological sciences tutor at Balliol since 2004, it says on my screen here, when he relocated from London up to here. And he is now the Professor of Evolutionary Developmental Biology in the Department of Biology in the university. And his work is very much on evolutionary biology, developmental biology, and lots about cells, which is what we're going to hear about this evening. Seb will speak for about 40 minutes, sharing his screen to show his slides. And at the end of that, I will come back in order to lead a discussion through the remainder of the event. We expect to finish in about 75, 90 minutes time. You've probably had explained to you that you place your questions in the chat box on the screen, and out of that I will then ventriloquize your questions and present them to Seb. I will perhaps merge together some questions if there are questions that follow similar courses. But it's an opportunity for you to ask Seb whatever the next 40 minutes put into your mind in terms of things you want to talk about. And I'm sure that it will be something that will provoke quite a lot of thought in people. So I look forward to plenty of questions. So I'm going to disappear by the magic of Zoom technology now and let Seb speak about his research. All up to you, Seb.
Thank you very much, Martin. So I'm just going to share my screen so that you can see my slideshow. There we go. So that should be in place now. So thank you very much for joining us today. Thank you for coming to listen to me talk about my work. My research sits broadly at the interface between evolutionary biology, genomics, and developmental biology. So developmental biology is the study of the mechanisms by which embryos build the adult shape and form. And I've worked on quite a number of different things over the years. But for today, I'm going to talk about nervous systems, and particularly vertebrate nervous systems. So what you're looking at here are two vertebrates, relatively late stage embryos that have been stained to show you their nervous systems. So vertebrates are the group of animals that includes us and includes other mammals, reptiles, birds, amphibians, and fish. And we have by far the most complicated nervous systems of anything on the planet. And you can see that stained beautifully in these two embryos here, one of which is a chicken and the other which is a very relatively simple sort of fish called a lamprey. Now, I need to go into these nervous systems in a little bit more detail before I start talking about some science. So first of all, vertebrate nervous systems have two major components. There's the central nervous system. So if you can see my laser pointer, that's made up of the spinal cord running down the back, and then the brain as well, which isn't stained particularly well in this particular specimen. And then there's the peripheral nervous system, which is all this fantastic network of nerves spreading through the body. The peripheral nervous system takes sensory input from the environment around an animal, feeds it into the central nervous system. The central nervous system does the processing. It also includes where memory, so where experience is introduced into things, and then it decides on the output. And then the output is fed back out through the peripheral nervous system to the muscles so that the animal can behave. And these two components make up our nervous systems and the nervous systems of other vertebrates. But I think it's important starting point here is that it's not just vertebrates that have a central nervous system. And I need to introduce you a little bit of animal diversity now. So here's a broad slide of lots of different animals. Vast diversity here, lots of different shapes and forms. Pretty much all of them have a central nervous system. And so if we're going to understand nervous systems, we need to start in the context, not just of vertebrates, so that's our own group of organisms, but generally across the animal kingdom. Now, these are animals. I'm sure you all intrinsically recognize most of them as animals. Perhaps not quite all of them. Some of them are a little obscure, but the vast majority, I think you would say, oh, yes, that's an animal were you to see it. But that's not a good enough definition for a scientist. And as scientists, we tend to use something a little bit more specific. And that's based upon evolution and phylogeny. And it gives me an opportunity to introduce some important concepts to you about how we depict evolution in forms of trees. So when we think of an animal, of a phylogenetic or taxonomic group, we usually use this sort of definition. So the descendants living and dead from a particular point. For animals, that point on this tree over here would be here, encompassing all of the animals living and dead, which is what this little grey triangle is supposed to represent. Within this tree, time is implicit from the past over here through to the future now up here. And these points here, these little things, we call these nodes, and these represent divergence points. So these are points where lineages have separated, in this case, this tree, a long time ago, and their descendants are no longer interbreeding, have gone on to evolve into very different forms of animals. What that means is, for something like this, is that when we see two lineages together in the tree like this, it means they're more closely related to each other than they are to anything else. And that's going to become important in a bit. Now, when we look at the animal evolutionary, this tree here, most of the other organisms on the planet are single-celled organisms. So bacteria and a variety of single-celled things like protozoans. But one thing that happened at this point in the evolutionary history of animals was the evolution of multicellularity. Animals are multicellular. Now, they're not the only multicellular lineage on the planet. Plants and fungi and a few other groups evolved multicellularity as well, but they did so separately. This, in our context of our lineage, is a unique evolutionary event. And you can see that multicellularity formed through the process of cell division and development. And you can see that going on over here in this developing amphibian egg, with the cells dividing but staying together to make this multicellular structure. But I think it's important to understand that animals are not just multicellular, but those cells are organized. Now, what I mean by that is that cells have to take on very specific functions to build tissues, to build organs, and for those tissues and organs to be integrated to form a functional, physiologically appropriate adult structure. And that's what gives rise to these amazing forms that we have in this picture here that I'm sure you're familiar with from the world around you. And it's the process of embryonic development that builds those adult forms from the embryo, ultimately from the single fertilized egg cell. Now, when we look across this group of animal forms, most of the ones on this slide have a central nervous system, but those ones that have a central nervous system have something else as well, and that is bilateral symmetry. So just to draw your attention to this side of the slide at the moment, most animals on the planet are organized with two definitive axes, a head and tail axis, we usually call it anterior and posterior, and one from the top to the bottom that we normally call dorsal and ventral. And this gives those animals bilateral symmetry, like this fish here, or this praying mantis over here. All the animals that have bilateral symmetry also have a centralized nervous system. So that includes most of the things that you'll be familiar with, the vertebrates such as us, insects, crustaceans, worms, in fact, most other animals that are alive on the planet today. However, it doesn't quite include all animals alive on the planet. There are a small number of groups of animals which don't have a bilateral symmetry and don't have a centralized nervous system. So that includes the sponges over here, which lack any definitive axial organization of their bodies. And then groups like the jellyfish and sea anemones down here, which have a definitive axis running from this case from the bell up here down to the tentacles, but they only have one axis, and that gives them radial symmetry. So if I put these three groups together into the phylogenetic tree, so that's effectively taking this little animal grey thing and blowing it up so that we can see the relationships. You can see that the bilateral animals are all together, so they're all more closely related. And then the jellyfish and their relatives and the sponges are progressively more distantly related. And what we infer from that is that the organization of cells along these axes, to give bilateral symmetry, has evolved progressively through time. Now, how does that happen? Well, to go into this, I need to talk about something else, introduce you to another idea, and that's the concept of signaling and how signaling is used by embryos to control how cells turn into the right sort of cell with the right function in the right place. So I want you to imagine a field of cells. These little hexagons here are my highly conceptualized version of a group of cells together. And currently, they're all the same color. They're all identical. How do we generate pattern in here akin to organization in an embryo? Most frequently in animal development, that's through the result of a signal, and the signal specifically coming from one end of the field of cells. So all it takes is for these cells at one pole of the field to become different and start to make a signal. A signal is normally a protein, so it's something produced by those cells and which spreads across the field of cells. That spreading process means cells close to it receive high concentrations of it, and ones further away, progressively lower concentrations. And they can use that strength, that concentration, as an indication of their position along this axis of the field of cells, and so can change their identity into different cell types accordingly. In this case, I've represented that with different sorts of colors. And it turns out that work from not just my group, but many groups over the years has identified the signals that control how axes develop in vertebrate embryos. So as a starting point, we know that all bilaterian embryos use a signal from the posterior end of the animal called WNT. We normally say Wnt for this. And that signal spreads from posterior to anterior. So the area that gets the most Wnt becomes effectively the tail end or the posterior end. And the end that gets the least level of Wnt becomes the head end. That's shared by all bilateral animals primitively. So we know it evolved a very long time ago. In fact, it's not just shared by these animals, it's actually also shared with their next nearest relatives, things like jellyfish and sea anemones that also use Wnt signaling to call the axis, but they use it to control this axis through here, from the bell to the tentacles. So that's the anterior-posterior axis, and that means effectively the head in these different groups of organisms is the same thing because it evolved from this common ancestral organization. So the second definitive axis I mentioned, the puff on this fish here, is this one here from top to bottom that we say dorsiventral. And that's where the nervous system, how the central nervous system is organized. But there's actually quite an important difference between an animal like this, a fish, and other vertebrates, and an animal like this, an insect, and most other invertebrates. And that is that the central nervous system is actually in quite a different place in these different groups. So in all vertebrates and a couple of invertebrate groups I'll come on to later, the central nervous system is dorsal, so it's on this side of the body. In our case, it would be running down our back, of course, and buried in our spine. We're kind of a bad model for understanding this because the human lineage, of course, is evolved by pedality, so we're standing upright. But primitively, we would have been horizontal to the substrate like this fish. If we take something like this insect, central nervous system is ventral, so it's actually running down the belly. And it's not just the central nervous system that has this kind of upside down organization. Some of the other internal tissues do as well, which gives this slightly bizarre situation where the animals look very similar from the outside, but some of their internal organization is upside down. Now this is not a new observation. It was actually first identified over 200 years ago by the French zoologist Etienne Geoffroy Saint-Hilaire. So through his work dissecting lobsters, which he almost certainly acquired in the Parisian fish market because that would have been relatively easy for him to do, he noticed this upside down anatomy. And he proposed it as a sort of a correspondence of a commonality of organization that he called the commonality of types. You have to remember that this was, nearly 30 years before the publication of Darwin's The Origin of Species. So while some ideas of evolution were around at the time from the work of Lamarck, the concept of shared ancestry wasn't prevalent in the scientific world. And a great many of his contemporaries took exception to that, including Georges Cuvier, another very prominent member of the Parisian science community at the time. And he took exception to the point where they had the kind of the 1830s equivalent of a Twitter war with a series of public debates on this issue, during which Cuvier effectively thoroughly dismantled Saint-Hilaire, primarily through the use of ridicule, so not that dissimilar to modern day social media in that context. And this idea that there was similarity in the organization, but they were upside down compared to each other, effectively went away for nearly 200 years. But of course, I wouldn't be telling you this if it wasn't back. And it's back because modern biology has identified the signal that's responsible for controlling the pattern and where the central nervous system forms. And it turns out it's the same signal in both of these groups of organisms. This time it's a signal we call BMP. And BMP works to control where the nervous system forms in both these groups of organisms. So in vertebrates like us, it signals from ventral to dorsal. So ventral is high and dorsal is low, and the central nervous system forms in the low part of the BMP gradient. Conversely, in invertebrates like this, the BMP signal goes from dorsal to ventral, and the nervous system forms in the ventral part, so again in the area of low BMP. So they really do seem to be sort of inverted with respect to each other. Now, these organisms are differentiated across their anterior, posterior, and dorsoventral axes, but they also have a third axis. In fact, if you differentiate any three-dimensional object across two orthogonal axes, you will create a third axis which is bilaterally symmetrical. And that's what all of these organisms are. So you can see this beautiful axis of bilateral symmetry running down this fish here. And all vertebrates have this. But as you're probably aware, while there's bilateral symmetry, left and right are not true mirror images of each other. Now I first became aware of this quite a long time ago now, when I was around the age of 12, when I first encountered this fellow here. Now, if this was a live lecture, I would be asking you to identify who this was. And when I gave this lecture live to those who were there at the Balliol Society on Saturday, I was very impressed by the number of people who knew that this, of course, is the Bond villain, Dr. No, and even a small number of people who could name the actor playing the part, so Joseph Wiseman, the Canadian actor. Now, why he's important here is that if, like me, you've been a geeky 12-year-old and read Ian Fleming's books, you would have found out that Dr. No once survived an assassination attempt because the assassin decided to target his heart and so shot at the left side of his chest. Because, of course, we are internally asymmetric. But what's important here is the assassin failed. And he didn't fail because he missed the left side of his chest. He failed because Dr. No had a condition called situs inversus, where his internal organs were mirror imaged, were effectively fully swapped over. So he had his heart on the right. It's not just the heart, which is asymmetric, it's all the other internal organs as well, the lungs, the liver, the pancreas, the spleen, the folding of the gut. All of that is asymmetric, and all of it is mirror imaged in situs inversus. And situs inversus is a genuine condition. Relatively rare. They say 1 in 10,000 to 1 in 20,000 people. But what's important here is it means that knowing that internal asymmetry can invert like that tells us it's genetically controlled. And we now know how that works. And it's down to a third signal. And that signal is called nodal. And nodal signals on the left-hand side. And what it effectively does is make left, left. And the side that doesn't have nodal signal becomes right, okay, develops into the right-hand side. And it's really important those two things happen so the left and right sides can develop and connect up properly. You can think about the plumbing issues if the left and right sides of the body don't, and what happens to all your internal tubes if they're not properly connected together. So it turns out that nodal signals make left, left, not just in humans and their closest relatives, but in all vertebrates. So whether you're dealing with birds or reptiles or amphibians or fish, nodal makes left, left. It's the left-sided signal. But of course, most invertebrates are bilaterally symmetrical and have a left and right sides too. And actually, while they might look superficially symmetrical, when we look inside them, we very often find subtle asymmetry, so subtle difference between the left and right sides as well. And as we showed and others showed some years ago now, that too is controlled by the nodal signal. So like the BMP signal and the Wnt signal, this is a deeply conserved, very ancient way of telling left from right. However, there is a difference. While vertebrates like us and a small number of invertebrates use nodal to make left, left, all the other invertebrates use nodal to make right, right. So they have nodal on the right side of the body, and that's what gives it its right-sided development. The side that doesn't have nodal takes on left-sided development and left-sided properties. So a bit like BMP, we've got the same signal, but it's inverted. Now, what that does, of course, is give us a clue as to why this is, why BMP is upside down, why Geoffroy Saint-Hilaire may be right, and why nodal is the other way around as well. And the answer comes down to thinking about these three orthogonal axes. So the very concept of an inversion was wrong. This isn't an inversion because an inversion would only change one signal. What it is a rotation. If you rotate this three-dimensional signaling model around the Wnt axis, you invert BMP and you also invert nodal. And the reason we think of these things as upside down is not really to do with biology at all, or the basic biology of the animals at all. It's more to do with us, in fact, and our nomenclature. The animals really aren't that different in the way they're working. What has happened is that at some point in the dim and distant past, lineages have separated, giving rise to the ancestor of vertebrates and some others, and the ancestors of most invertebrates, somewhere deep, deep in bilateral animal evolutionary history. And what has happened is that one of those lineages has taken on a different orientation to the substrate than the other. Effectively, this is because our wording, our nomenclature for dorsal and ventral doesn't come from something intrinsic to the biology. It comes from how the animals orientate themselves with respect to gravity. So we think of the dorsal side or the back as upwards with respect to gravity, and the ventral side, the belly, as down with respect to gravity. Of course, the animals don't know that. It simply happened that sometime dim distant past, and we're talking perhaps 500 million years ago, one of those lineages took on a different orientation to the substrate. And the mistake made by Cuvier and those that followed him was thinking that we were comparing, to understand this, we should be comparing a modern lobster with a modern vertebrate. We're not, we're thinking about a relatively simple ancestor that would have almost certainly have had a relatively simple worm-like body plan. Now, so that gives us a framework for understanding animals, and it allows me to move into the second part of my talk, which is really thinking about vertebrate nervous systems, because it means that nervous systems are homologous across these groups of animals. What that means is they've definitively shared in the sense that they evolved from a common ancestor with a centralized nervous system in the dim and distant past. So I'm going to return now to nervous systems, to my picture of the chicken nervous system and the lamprey nervous system. I'm going to move a little bit more now into some of our more recent work in trying to understand how this degree of complexity evolved and how we can use developmental biology and genetics and comparative biology, so our understanding of evolution, to gain insight into where this incredible complexity has come from. So to do that, I need to dig a little bit deeper into the animal evolutionary tree. So I'm going to go back to my phylogeny over here with the bilateral animals and the two outgroups. This obviously is a massive simplification of the diversity of animals on the planet. And if I take a more up-to-date tree from a research paper on this, not one of mine, this is from a published paper, and show you the relationships of the major animal groups, you can see there's a large number more than I'm depicting on here. Now, I'm sure you're probably trying to read through those names and thinking, I've never heard of half of these. And that's not surprising. Even most professional biologists would not be aware or not be particularly aware of this level of diversity. Fortunately, for today's talk, we don't have to worry about the vast majority of the lineages on here, because I'm going to focus us right down on one animal group, and that's the animal group that contains us, contains the vertebrates. And that's down here in blue towards the bottom of the tree. It's called the chordata or the chordates, and that's our phylum. So that's the upper taxonomic group that we as vertebrates sit in. And if I blow that up into a more friendly, simplified tree, we can see that the vertebrates sitting down here, so this range of things that we're related to, are related to two other groups within the chordates. They're called tunicates and cephalochordates. These effectively are our sister lineages. They're as vertebrates, they are our closest living invertebrate relatives. They both have dorsal nervous systems like we do as vertebrates, but they're much simpler than the really complicated nervous systems that we see down here. And what that tells us is that at some point in the evolutionary history of the vertebrates down here, a great deal of extra neural complexity evolved. Now, complexity or complicated, that's a term I've been throwing around quite a lot. I think we need to explore a little bit about what it means. There are some really good, crisp scientific or mathematical definitions of complexity. But as a practicing experimental biologist, they're a little too esoteric to actually prove testable in the context of laboratory work and experiments. And so what we do is to break this aspect down, this overall concept of complexity down into a number of simple subcategories to provide simple testable questions that we can then examine. And doing that now, I'm going to introduce you to three today. So by more complicated, could we mean there are more cells? Certainly vertebrates are bigger than most other animals, and their nervous systems are bigger. They must have more cells in. So does more cells equal more complexity? Almost certainly yes, but where did they come from? Could we mean more intricate patterns of cells? If we have lots more cells, or do we need to organize them better? So perhaps we might have more intricate signaling to create more intricate patterns to give a higher degree of organization. Or do we mean new types of cell? So I haven't really spoken much about what a cell type is as yet, but I will come back to that at the end. So if that concept's not really clear to you, I'll try and clarify it in a little bit. And of course, underlying all this, we really want to know how. It's not just what the difference is, but how did that difference come about? What's the mechanism that generates this complexity, and how has that changed? And I'm going to introduce a little bit of work at the end about the genetic mechanisms to control these and how they might have changed. So the starting point of this, I need to introduce you to one of the systems that we work with experimentally, and that is the spinal cord. So much of my work over the last sort of 15 or 20 years has focused on developing spinal cords. So I'm sure you're all aware of what the spinal cord is, that bit of your central nervous system that runs down your back, embedded inside your vertebrae for protection. It's simpler than the brain, considerably simpler than the brain, but this anatomical diagram of a human spinal cord shows you it's still a pretty complicated structure with lots of different bits to it, all doing different things. And these different tracks and the cells within them are all necessary for the correct functioning of this bit of the nervous system, for receiving sensory input, for feeding information to the brain, for getting information or instruction back from the brain, and making things happen by controlling muscles. So the details of all of these aren't important for today's talk, but one thing on this diagram is important, and that's this little thing in the middle here. And what this is a hole. So fundamentally, topologically, the spinal cord is a tube. Now, the hole looks pretty small in this adult version. It is pretty small, although it's still pretty important. This is the site, for example, for which cerebral spinal fluid is based, in the hole in the middle. But I'm going to conceptualize this now and take it back to embryos to show you a little bit more about how this tube structure is important in generating this complexity through cell number and through pattern. So over here, we've got an embryo spinal cord. So it's a little bit simpler than this complicated diagram down here, but you can see it's still got quite a large number of cells in it. This is from a chicken embryo, relatively early in development. And the hole in the middle is a little bit closed up here, squashed up in this preparation, but you can see it's much larger. So I'm going to abstract that now as this sort of structure, just a glorified diagram of a tube. Now, if you think about all the different cells and structures which are forming here, this requires lots and lots of differently specialized cells to perform these different functions. And we know a great deal about where they're organized during embryo development along the spinal cord. And it turns out they are extremely intricately patterned. So again, I apologize for the complexity here. I think it's important to show that for you to know that there are really quite a lot of different things going on in even a very early embryo developing spinal cord. We have all these different sorts of cells. That thing, they take on all sorts of different functions. They're all necessary for the correct functioning of the spinal cord for it to do its job. But I think the important thing here is that they're so precisely orchestrated or organized into this intricate pattern, which is running along an axis from the top here to the bottom down here. And that, of course, should be a concept that's familiar to you, organizing cells along an axis, because it's very reminiscent of how the overall embryo is organized. And it turns out that there's another similarity as well, and that is that this pattern here is controlled by signaling running along this axis. And work by many groups over quite a few years has really worked out what those signals are and how they work. And it turns out two of them are familiar. So from the top end here, we have two signals working together this time, BMP and Wnt, the same ones we've heard of before. Now, it's important to emphasize here that the reason these same signals can be doing a very different job in a spinal cord compared to what they were doing in an early embryo to control anterior, posterior, and dorsal axis is down to time. The axes, the overall embryo axes, are set up very early in development. The spinal cord forms a little bit later. Those signals are no longer needed for controlling the early axis, so they can be reused to control, in this case, the patterning within a specific organ system within the body. So it's obviously different in that both signals are coming together. And there's another difference as well, in that there's a third signal called HH that comes from the other end. And these two opposing fields signals divide up this whole field of cells to generate this really intricate pattern down here. Now, one simple hypothesis for how we could have got more complexity in vertebrates is simply introducing more complexity in signaling. So as a very simple level, does more signaling mean better control, more intricate patterning, more complexity in the nervous system? Well, that's something that we and others have looked a great deal at over the years. To give you a little insight into the structures of the spinal cord regions of these animals, I'm going to use some very simple line diagrams. So vertebrates are more complicated and they have a lot more cells. I'll talk a little bit about why those cells are layered up here compared to up here in a minute. And these two groups of organisms, our closest relatives, have much simpler single-layered spinal cords. So could extra signaling mean more complexity here? It was an attractive hypothesis, but the answer turns out to be no, because both of these lineages have all three signals in the same place. So effectively, they're the same. They have exactly the same signaling environment. Even though there's relatively few cells and a tiny number of cells in this lineage, they still have those same signals propagating across those fields of cells. So it's not extra signaling, but it doesn't mean that the signaling isn't important. And we know that from experiments. And what we and others have been able to do is to use modern genome editing technology, things like CRISPR, which I suspect many of you would have heard of. It's been a very potent and powerful new tool for understanding gene function in biology. And CRISPR, applied to this sort of question, allows us to manipulate the function of the genes making the signals and work out that they are important. They do have an impact, but they're not having the same level of impact. So I couldn't resist putting these on here. So these are real data for these sorts of things. I'm not going to make any attempt to explain these to you. It would take me hours, and I think Martin would get very unhappy with the length of time I was going on for. But I thought it's important you get to see what actual real data look like in this sort of field and how complicated and intricate it can be. What I'm instead going to do is just try and conceptualize the outcome of this sort of experiment so you can get a feel for what the evolutionary or developmental difference is here. And I'm going to use this diagram as a basis for that. So this is my simple signaling diagram, this time turned on its side from earlier when I was explaining how signaling works. So if we imagine this as a kind of model, in vertebrates, this is a bit more complicated because we have signals coming from both ends, and as a consequence, we end up with cells divided into more intricate patterns based upon the number of signals. When we compare that to these outgroups, so to these sister lineages, which lack that neural complexity, what we find are the signals are still present, but the response in the cells is different. So the cells are not reading the signals in the same way. And in fact, what's happening is rather than reading the signals as cohesive populations of cells generating this sort of pattern, individual cells are responding and changing their identity, but not in this sort of coordinated way. So something has changed in the downstream, in the mechanism downstream of the signal. We actually know quite a bit about what that is, but I'm not going to go into that today. It turns into some relatively complicated developmental biology and genetics. And I think it's the basic point is the change is internal to those cells, is down how those cells are reading the signal. Before I go into any more on this, I need to step back a little bit and think about the second question, which is cell numbers. So a spinal cord has a very, very large number of cells in it to perform all these functions. And that's why I've drawn it in this sort of way in this diagram here. many, many more cells compared to the numbers of cells in these two outgroups. So the number of cells, the size of the organ, the size of the system has definitely increased along with the intricacy of the pattern. Now we know how those extra cells arise, and it's down to a stem cell population. So a stem cell is a type of cell that can make it, that can renew itself, but it can also make cells that can go on and do something else. And in the spinal, the stem cells sit at the middle next to the hole in here, and they keep dividing for a very, very long time during development. Some of them stay as stem cells to keep that population going. Some of them move slightly laterally like this and differentiate into neurons. And because this goes over a long time, it kind of builds up layer upon layer upon layer of cells. So you get this sort of layered complexity to the nervous system. And this is controlled by another signal. That signal is something we haven't heard of before. It's called Notch. I apologize for the names here. The names are buried in, the reason for these names are buried in genetic history. So they weren't named in a way that's sort of modernly appropriate, but the signal is embedded in the literature in scientific knowledge as called Notch. Now, when we move to look at our outgroups, then we find both of these things are absent. So there are no stem cells in these two. The stem cell population that lies in the middle of the neural tube, in the middle of the spinal cord, is something which has evolved in this lineage here. And secondly, the signal that makes them, that keeps them going, the Notch signal, is also missing. So we can see some of the evolutionary event which has led to this sort of increased number of cells in the spinal cord. And we can build those two together because it turns out that not only does complexity come from changes in the intricacy of the pattern and changes in the number of cells, but these two are related. And that's because this process over here, where intricate pattern is forming downstream of the signals in vertebrates, is actually happening in the stem cell population in the developing spinal cord. So it's these stem cells which are susceptible to the signal which is doing this. So probably the combination of these explains how we've been able, how the ancestral vertebrate was able to go from a relatively simple back ancestor like one of these two to something much more complicated with many more cells organized in these precise patterns. Of course, they're not just organized in these precise patterns. Alongside this, we think there's also a third type of evolution going on, and that's the evolution of cell types. So when I've been showing you this sort of diagram here, I've been using colors to represent different sorts of cell, but I think I need to explain biologically what that means now. So when cells divide in an embryo, as they've been doing over here, every daughter cell inherits an entire copy of the genome. So they're all genetically identical. So all the cells in this field, all the cells in this embryo, have exactly the same genome within them. Now, a genome of a typical animal, like us, contains around 25,000 genes. And every cell has all of those 25,000 genes in. So what actually makes them different if they carry the same DNA? The answer, of course, is not what genes they have within them, it's which ones they're using. We would say expressed. So if you're familiar with the sort of central dogma of molecular biology, it's the genes which are being turned into RNA and which is then being turned into protein, and protein being the effector molecules of life. So those are the genes which are important. And if we think of cells which are forming muscle compared to, say, cells which are forming skin, the muscle cells are going to express the subset of genes they need to perform that function. So the things they need to make to be a muscle cell, and they won't express the ones that a skin cell needs, and vice versa. A skin cell will express a skin cell specific set of genes, but won't express the muscle genes. Now, this is something we and others have been working on for a great many years. It's been laborious work because of technological limitations, essentially working on a cell type by cell type basis, trying to work out what was going on inside the cells and trying to understand how they were forming. What's changed in the last few years is technology, and particularly the rise of what's called single cell biology, which allows us to not just see what's going on inside a cell in terms of what genes it's expressing, but to do that simultaneously from thousands or even millions of cells at the same time and ask, what genes are expressed in all of those cells at any one point in time. Now, this sort of research, really driven by the development of certain sorts of technology and high throughput sequencing, gives us very, very large data sets and very, very high dimensional data sets. I'm not going to talk about how we analyze these at all. The outcomes look something like this. Again, this is probably too small on your screen for you to even see the details, but these are thousands and thousands of individual cells which have been analyzed to understand how they relate to each other. And then we can then ask how cells of different sorts relate to each other within a species. And then we can even take those data and start to compare them between species, so we can compare one sort of cell to another sort of cell in different species and understand how they're related to each other in terms of the genes that they're expressing and the functions that they have. So this is identifying lots of different relationships between different sorts of cell and even really distantly diverged species. So these comparison here are two groups of organisms which are separated by at least 500 million years of evolution. I'm not going to go into any detail on the majority of this. It gets very granular and quite descriptive, but I do just want to pull out a couple of really interesting things that are coming out of these sorts of high throughput systematic comparisons of cell identity between species. And one is a case study, a case example on a particular cell type called oligodendrocytes. So you may have heard of something called myelin. Myelin is a fatty material, and what it does is it insulates our nerves and sort of is wrapped around the axons, the parts of our nerves, like the plastic around the cabling, like electrical cabling. And that insulation is really important for accurate and long distance transmission in nerves. And when it gets damaged, then it can cause neural diseases. Now, myelin is made in vertebrates by a cell type called oligodendrocyte, but it's something that's specific to vertebrates, something that our lineage evolved that other lineages don't have. And through these sorts of cell comparisons, we've been able to identify where those cells come from, so where they originally evolved from, but not just that, to start to work out the specific genes that have changed their expression to drive the development of these new cell types and provide these kind of new properties to nervous systems. So the final couple of minutes of my talk, I'm going to step from this into something a little larger, which is a more systematic understanding of how these sorts of molecular changes can generate complexity. And I need to introduce one more concept to you now before I wrap up, and that's the concept of genome duplication. So we all have a genome, but by comparing the genomes of different lineages, we can work out when major evolutionary changes have happened. And one of the things that's been known for more than 20 years now is that the common ancestor of vertebrates, right down here at the base of the vertebrates, underwent 2 rounds of whole genome duplication. What that means is that the genome in some way doubled and then doubled again. Now, as I said before, the typical animal genome has roughly 25,000 genes in it. That would give 100,000 genes, but not all of those genes have been kept. In fact, the vast majority of those duplicated genes were then lost. And that, of course, is essentially a sort of a truism in evolutionary biology, the sort of use it or lose it. If you have multiple copies of a gene, then if they're doing the same thing, evolution will almost certainly lead to one of those copies being lost, or all but one of them being lost. And that's simply because they're not necessary, and evolution doesn't keep things which aren't necessary. So most of them were lost, but not all of them. Some of them were kept. And what we've been able to do by comparing single cell data from lots of different species, and this is from several different species, and this is particularly focusing on brains, is to ask, what are those new genes doing? And this has identified 2, I think, are really important things. One of which perhaps is a little expected, and that is that these duplicated genes, which we know evolved at this point here, are much more likely to be contributing to cell type evolution in vertebrates. So we think that they're responsible in part for this big burst in complexity of cell types that we know occurred at this point on the tree. We expected that that would be the case. In fact, we've long suspected that this would be that the duplicated genes that evolved this time would be important for these processes. But it's been incredibly hard to prove until we had these sorts of data, and we think we now have that. We now have that solved. The second outcome, which is perhaps a bit more surprising, is that those duplicated genes didn't just do this and then disappear. They've been kept. They've been kept by all subsequent vertebrates. And all subsequent vertebrates haven't just stood still. They're continuing to evolve their brains. Brains have diversified. A lizard brain is not like a mammal brain. A bird brain is different to a crocodile brain. There's evolution going on all the time. And we don't have enough data to fully understand this. But what it looks like where we do is that those genes are still contributing to cell type evolution through modern vertebrates. So it looks like that kind of burst of innovation 500 million years ago not just gave us the ancestral big brains, but have facilitated adaptive changes, evolutionary changes in brain complexity for a very long time afterwards. Okay, so I'm going to wrap up there. So I just want to finish on an acknowledgement slide. So it's really important to me to acknowledge all the people and organizations that help. So we couldn't do this work without funding, without money, and I acknowledge the major organizations that help fund us. This is my current research team, and I'd particularly draw your attention to Huan Zhen, whose work on cell type evolution I talked about right at the end. So he might be online today. Actually, he's a Balliol graduate student, and then also the very many people who have helped me, who have worked with me in the past, who fantastic people who I have done a huge amount with me over the years and are responsible really for actually doing a lot of the experiments and work that I've talked about today. So I'm going to finish there. Thank you very much. I'm going to stop the screen share.
That's great.
Martin, over to you, I think.
Indeed. Yes, thank you very much indeed, Seb. That's fascinating. And, it seems to me that a lot of this also goes back to something you talked about in the first 2 minutes of your lecture, which is how those divergences just happen. I mean, with my primitive historian's idea to sort of back in the mists of time sort of thing. And so I want to ask you about that, while perhaps I also want to encourage everybody else to pose their questions. I misspoke at the beginning by talking about putting questions in the chat. You have to put it in Q&A, which is probably pretty self-explanatory at the bottom, and I'm pleased to see there's some there. But everybody do take your opportunity to put your questions in there, and I will then pick them out. But in the meantime, Seb, how do we deal with this? This sort of the origins of all this lie in divergences that happen. Yes, sure, but why?
Yeah, so there so we don't know exactly how divergences that occurred hundreds of millions of years ago happened, but we do know pretty well how they've happened in relatively recent history, evolutionary history, and we can be pretty sure that similar mechanisms would apply. Essentially, one of the major ways that lineages diverged is through geographical separation. So if you imagine a species that's living on an island, if it manages to colonize a separate island, great, population can expand there. But those two don't meet anymore. They're separated off, and so they start to go their own evolutionary way. So that's an island in the sea, but an island could be any separation. It could be a mountain range, it could be an ice field, it could be ocean currents that no longer mix different groups of sea creatures together. And once you have that separation and you lack interbreeding, then those lineages are free to diverge. And if they're in different places, then they'll experience different selective pressures and they will start to change according to their environment, according to what's fittest for the particular environment at the time.
Right. Thank you. There's a couple of questions from Nick Wilcox, who thanks you very much for your talk and says that it's fascinating for someone to hear after first hearing about these problems more than 60 years ago, which is quite humbling for the rest of us. So one question he asks is, and if I get this wrong, tell me off, how are primary cilia involved in left-right determination in invertebrates, regardless of which way round they develop?
That's a great but very specific question from someone who knows a little bit about how this works. So
Okay.
primary cilia are, so we know that in some vertebrates, cilia, which are these tiny organelles that stick out of cells, are involved in the initial breaking of left and right. So we've studied this, and cilia are also involved in some invertebrates, but not all. And it turns out, I think, that cilia are probably a manifestation of molecular structures which are necessary to break left-right asymmetry, but not essential. And that molecular structure is something which is chiral. So cilia are intrinsically chiral, which means they have circular directionality within them. And that means it can be used to
Okay, tell
break
me a bit more what chiral means for those that
chiral. It's got circular orientation.
Oh, okay, right.
So it goes round and round inside in a particular direction. And that's really important. And effectively, it's a molecular asymmetry, which is being read into the asymmetry of the embryo and hence into the asymmetry of the adult. And there are other ways of doing that involving molecules that are similar in structure, but not necessarily in cilia. So yeah.
Go on, tell me a bit more about that. What would be those different molecular structures?
So in snails, we know
Mhm.
that it's based upon some of the cytoskeletal elements. So cells have a skeleton within them, and those skeletal elements are circular as
Yeah,
well. And so it's based upon those.
great. Thank you. Nick has another question, which is, can you see how many neuronal types evolve gradually via useful intermediaries, such as in early vertebrates? So in
So
early vertebrates, these neuronal types
yeah,
evolve
yeah.
first.
So it's a good question as well, Nick. So we can see some of that. I don't think we can see it all. Some of that might be limitations of the data in that we just don't have
Yeah.
enough data yet. Some of it may be just inevitable limitations because some of the organisms which underwent these processes have become extinct. So there's now a big gap in complexity. So if we had more lineages that were separated that carried different levels of change, we might be able to see more of that. But I think because we're restricted to what's managed to survive for this sort of molecular genetic analysis, I think there might be gaps that we simply can't fill in there.
So you can't count them
No, not all of them. No.
because the data is not insufficient, right? So
Yeah.
it's a not you haven't got a complete set. Yeah,
No,
not a
no,
complete
and we
data set.
possibly won't get a complete set, I think.
No. Right. Anthony Cluder wins a prize for asking a question which occurred to me too, which is, could you tell us what a signal is? Is it electronic or chemical? Perhaps a bit of both. And how do you find it? And how does it act on the genome DNA?
Oh yeah, it's a lot of questions. So the majority of the signals are chemical. They're made of protein. They're actually proteins which are secreted by a cell. So just like
Okay.
most of our bodies made of proteins, but these specifically are pushed out of the cell. They don't have to be, so they can be other forms of chemical as well. Electrical signals can operate in biology, but we don't have very little evidence for them doing this sort of role. So
This
although
is
we obviously,
inherently chemical. This is inherently biochemistry, as we would call it.
yeah, yeah, absolutely. So where's the rest of that question gone?
How is it? So,
Yeah.
it's
How
chemical.
does it
How is
found?
it found, and how does it act on the genome?
So the majority of these signals were originally found from early mutagenesis screens carried out in invertebrates like Drosophila. That means that Drosophila, strange-looking Drosophila were found where development was going wrong. And when the gene which was mutated was found, it was turned out to be encoding one of these signals. So the majority of them were found in that way from our experimental model systems. How does it act on the genome? Typically, one of these signals will be recognized by a cell surface receptor protein. So there's another protein that specifically recognizes the signal and sits on the surface, and then that relays a signal. It's kind of a bit like a sort of a beacon on the outside of the cell, which catches hold of the signal, and then the inside of that beacon
Okay.
is inside the cell, and that
Yeah.
tells the nucleus to do something. So they don't all work like that, but that would be the most common mode of action.
Why do you call this signaling?
Well, because information is going from one cell to another. So it's a form of information transfer.
I mean, or a form of communication, yeah?
Yeah. Yeah.
So Aaron Holden says, how do the signals spread by diffusion cells playing past the parcel, moving it from one to the other, or is there some bulk fluid flow of proteins through that take the signals to a whole series of ones at the same time?
Okay, so there's three answers to that question. Some of them work simply by diffusion, but that tends to be the ones that aren't proteins. So small molecules can spread freely by diffusion. Large molecules like proteins, where we know they're moving in more complicated ways, sometimes attached to lipid molecules, possibly also wrapped up in little packages of cell membrane called exosomes. And the third answer to that question is for quite a bit of this, we simply don't know. So it's an
Mm.
extremely difficult thing to observe, signals in action. And it's quite possible that the same signal could be operating in all three ways in different contexts as well. There's no reason to assume that one is occurring somewhere all the time. They could be in different ways in different contexts.
Okay, which also I think probably links in with Matt Hitchens's question about is the signaling in the most basic animals more like the signaling across our body or the signaling within our spinal cord?
Um, so at the basic biochemical level, actually, it works in exactly the same way. So the signal is produced by one cell, it spreads by whatever mechanism, and it's received and yields a change. The differences are not in the way the signal sort of working, but in how they're being interpreted. In a sense, if you
Hold on,
think,
you'll have to say that again. Say that one sentence
yeah.
again.
So the signal is providing information, and that
Yeah.
information is how far you are away from something. What you do about that depends upon what you are.
Mhm.
So if you're a
Yes, got
neural
that.
tube,
Yeah.
yeah,
So most basic animals are never very far. The cells are never far away. Is that the
never
point?
far away, or their range of possible responses is limited.
Right. Okay. But they're not inherently different.
No, no, they're not. No, there's no reason to think that they wouldn't have the capacity to evolve some additional complexity if selection was appropriate. Yeah, but it would
Thanks.
require change. They respond in the way that's effectively programmed into the genome that they're able to respond.
Esme asks a question, which she apologizes, saying it could surely be phrased better, but it seems absolutely fine to me, which is how is signaling linked to gravity, giving rise to the axes, if an embryo is being moved in relation to gravity during its development? So the gravity should confuse everybody, but it doesn't, it seems.
Yeah, generally speaking, it doesn't. And that's because the signaling is occurring kind of within a packaged environment, so it's protected from the outside world. There is one or two examples where gravity does play a role very early in development, that have been identified, in terms of particularly breaking an axis. So working out which side of the embryo, say, a signal is going to start on. But beyond that, I think this is happening at a scale and an environment where gravity isn't really necessary. Remember, this is all wet, so we're not dealing with, air here. We're dealing with a
Okay, yeah.
complicated cellular environment with chemicals moving around within the matrix, within a cell, with it's basically an aqueous environment. Yeah.
So gravity is entirely external to this process.
It's there all the time, of course.
Yeah, but it's
But
not a
I
player. Sorry
yeah,
to use chaotic language here.
yeah.
OP Yadava asked, did God play any role in signaling? A
Well,
question you may decline to answer, but I think it's a very good question to answer, to ask.
so I mean I'm afraid as a scientist I can't regard that as a testable hypothesis. So no, I would pass that over to the theologians to answer.
Yeah, fair enough. Martha Rushbrook says, is it really that a handful of proteins are controlling organization so fully, or is the reality more complex? And I
Do you
think that
know?
might have some hint to the God question earlier in a way.
Yeah, it's
Is everything just coming from a handful of proteins in that sense?
not everything, no, and I've simplified a huge amount. So for a signal to work, there would be dozens of proteins involved. It's not just a signal and a receptor protein, but something has to help make the protein and export it and guide it and then catch the signal and process it. So there's more going on. But what has been a remarkable finding of developmental biology, but across these different animal groups over the years, is that a huge amount of development in all animals is controlled by actually a small handful of signals. And we've mentioned four or five of those today. There's actually only one or two more, which together explain the large amount of this sort of developmental patterning that's occurred, as far as we know, in all animals. I hesitate to say it's in all animals because, of course, we've only studied in depth a handful of things. But where we study them, we find that these same signals are involved.
Hmm. Joseph Muscat seems to go back to my question about why divergences happen. So why did the differences between ventral and dorsal organisms, so to speak, come about by chance, or were they due to evolutionary pressure? Is there something else going on that's pressing in on the emergence of these different trajectories?
Yeah, so again, that's a difficult question to answer precisely because we can't go back in time and observe what actually
No.
happened. But the, the most likely explanation is to do with the mouth. So we know that an evolutionary event that happened around about the same time was the evolution of a through gut. So something like a jellyfish or a sea anemone has a blind gut. It's got one opening, food goes in and waste comes back out the same hole.
Right.
Most bilaterian
I'm not
organisms
sure I
have.
wanted to know that, but yeah, right. Okay.
Well, this is biology. I'm sorry,
Yeah.
it gets a little messy at times. So bilaterally symmetrical organisms almost all have a through gut. So they have
Yeah. Okay.
two ends. The orientation of where the mouth is almost certainly going to be pointing downwards towards the substrate in an ancestral organism. So
Hmm.
one of the possibilities that separates these things out is separate evolutionary origins of a through gut and a mouth, and the mouth evolving in slightly in different ways in different lineages. So that's one
Therefore,
possibility,
creating the differences between
and
ventral and
therefore
dorsal.
creating the differences between the therefore effectively creating the rotation. Yeah.
Fair enough.
But this that's quite speculative. That's not. I don't regard that as something that's proven. There is some evidence that supports it, but I don't think it's definitive.
No, well, I think there's a great deal in what you've been saying, which actually demonstrates the sheer liveliness of the field you're working in, where a lot of
Mm.
things are coming to be known, but are still not known in any with any great certainty.
Yeah.
Um, Benjamin Sisman says, could you speak to some of the examples where gene duplication or amplification can lead to detrimental effects at an individual level, such as with MYCN transcription factor amplification in neuroblastoma cancer?
Yeah. And Ben's absolutely right that um, when sometimes if a single gene duplicates and you end up with multiple copies, that can have really difficult effects for development because and the reason for that is almost certainly to do with dosage. So you've got three
Dosage,
copies of one
new word.
gene, and
What,
you've
what do
suddenly
you
got
mean?
more of one product than you have of others. And sometimes for
Okay.
many proteins, they have to be in sort of the right appropriate amounts in a cell. So the distinction here with that sort of duplication and whole genome duplication, we think, is that whole genome duplication instantly doubles everything. And so while you
Mhm.
might have twice as much of everything, the relative proportions of proteins that are coming from those genomes stay the same. And so we think that that is actually much more permitted by evolution or by the system than the amplification of some single genes, like the example of the MYC transcription factor in this context. So it's about relative abundance, probably. And
Are we getting into a world where it's possible to work out what are the determinants of diseases happening?
yes, oh yes. Yes. I mean, I wouldn't say that was particularly what I'm doing, but so the medical genetics and molecular biology is understanding many of these things in quite some detail. Yeah.
With hopes of treatment?
Oh, I think so. Yes. Yeah. I
Good.
mean, medicine's advancing incredibly quickly. I think the limitation is becoming affordability rather than
Right.
the
Okay.
ability, rather than the future potential for medicine to actually treat many of these diseases.
That sounds a very Balliol answer. Yeah. Okay. Paul Shotton has a couple of questions, which I'll take as one, which is what caused the doubling and then redoubling of the number of genes you referred to? And have there been other instances of this happening? And can you explain more about the mechanism behind your phrase of use it or lose it?
Yeah, thanks, Paul. Yeah, so what caused the doubling and redoubling? So we can trace the ancestry of the separate genomes which are present, or the very ancient genomes which are present in our genome. And we know that the first genome duplication was down to a hybridization between two closely related species. And the second one was separate, or was slightly different. So we can trace that. So effectively, and it's something that actually happens pretty commonly. There are quite a lot of other lineages where genomes have duplicated. There are even some very recent genome duplications. A great example is salmon. So salmon have undergone multiple additional genome duplications on top of these ancestral genome duplications. The difference
I
is,
think you're using recent in a different way from how a historian would use it.
well, recent in the sense of millions of years. Yes. Okay.
Yeah.
Well, recent from my perspective, comparatively recent in terms of evolution. What's
So
different?
there's multiple duplications, and we don't really know what prompts those to happen.
No, we don't know fully. No, I mean it doesn't seem to be that difficult a thing for some groups of
Yeah.
organisms to do, particularly ones perhaps where there's external fertilization, and the possibilities for hybridization are higher between closely related species. Um, the difference is that the majority of them don't seem to be connected to this sort of bursts of innovation. So they simply sit there. And as far as we can tell, they're not doing anything particularly important in terms of evolution. Why that is probably down to the selective environment. It's not just about what you have inside you, it's about the environment around you that provides the selective pressure for change. Paul's second question was about use it or lose it.
You
Yeah.
use it or lose
So,
it. Yeah.
so this is the case that if you have two genes that are doing exactly the same thing, it doesn't matter if one of them gets mutated because the other one will carry on doing it. And so over time, one of them is bound to get mutated. And once it's mutated, it's lost its function. It's not there anymore.
Mm.
And the other one will carry on doing it. Of course, you then don't have any backup, but evolution doesn't have a kind of a forward look. So it can't see into the future and think, I might need a backup. I better keep that other gene around. It just goes because it's not necessary. Selection won't keep it. And so it disappears.
So there's a functional logic to these things.
Yeah.
I think Tegan Phillips's question has probably been prompted by what you just said about external fertilization. So for animals which develop embryos within a parent organism, that's like us, are there any differences between embryos developed in vivo versus in vitro, or does the lab environment fully correspond with the elements in a womb that would constitute the natural environment?
Yeah, yeah, that's a really good question as well. So actually the majority of organisms that I work with are developing externally. So
Right.
so these are organisms that have external fertilization. So that's not an issue. If you were doing for labs that do experiments with mice, say,
Mhm.
which is a very common model, then sometimes they would try to grow those very early embryos outside of the womb or outside of implantation. And yes, there will be differences. So the environment is important. It seems to be less important than changes in internal mechanisms in these
Yeah.
lineages. But yeah, I think it would be naive to think that the environment that something was actually growing in didn't have an impact upon the development of the
And
animal.
the thing that would have an impact would be what? The chemicals they are exposed to
The
externally?
chemicals, but the physical contact with other things,
Yeah,
possibly the level of nutrition or oxygen that was being provided by the environment. All of those
sure.
could have an
Yeah.
impact. Yeah,
Oxygen deprivation or something, yes.
yeah.
Hmm. Interesting. And Martin Jockin asks to go back to the folding of the neural plate to form a tube. You know, this was so central to what you were saying about the spinal cord.
Yeah, yeah. So what Martin is alluding to there is that the reason we have a tubular nervous system is it starts out as a plate of a flat sheet of cells, which then rolls up to form a tube, kind of circularizes by a
Yeah,
rolling
yeah.
motion we call neurulation. So we think that evolved around about the base of the chordates, probably a bit older, but we don't know how. So um, it's many people have thought about it, and nobody has come up with a really good explanation as yet. So cellularly, we understand how it works really well. So we know mechanistically it's very obvious how that
Yeah.
rolling occurs. But in terms of what would drive the evolution of a tube or as opposed to a plate, I'm afraid now, Martin, we don't know the answer to that.
Yes, there's quite a lot of sort of missing motors in all this story, aren't there? I'm not, it's not your fault. This is just that we understand that things happen, but we don't necessarily understand quite what is driving the dynamic of change.
Yeah, yeah. Well, I mean, we understand, yes, we understand that what's driving the dynamic of change in modern animals, but we
Yeah,
don't understand what's driving the dynamic of evolutionary change.
yeah,
Yeah.
exactly. Michael Popp asked briefly about the role of geometric space in protein signaling.
Ooh, yeah. I'm not quite sure how to interpret that question, actually.
Okay.
So
What do we mean
protein,
by geometric space in this context? I mean,
yeah,
it's
I
presumably the
assume
distance.
it means I'm sorry, Michael, I'm gonna have to try and interpret the question here.
Yeah, please do. Take it the way you make sense to you.
So I think by geometric space, Michael is alluding to the three-dimensional folding of proteins to create structures, to create three-dimensional structures and what they occupy. Um, and it is important in protein signaling because proteins, if they're going to interact, if two proteins are going to interact, they have to fold to form the correct structure so that they recognize each other, like a sort of a lock and key type interaction, so they
Okay.
physically interact properly. So it is important in that sense. And you tend to see the evidence for that in that proteins doing the same job in very, very different organisms tend to have conserved three-dimensional structures. But I'm not sure if that's quite answering the question, but I think I'll stop there because otherwise I'm going to drift off into parallel things that might not be relevant.
Fair enough. Anthony Cluder, who's asked a question before, has a question here at the end, which you said a mutated gene would be lost. What if it instead worked, but in a different way and took over? We're a bit in the Dr. No land here. And would that be the origin of cancerous cells?
Yeah, mutated genes sometimes acquire new functions, and that the organism can then use, and that's what we think has happened in the evolution of new cell types, and also probably in different ways of controlling pattern and the more intricate patterns. If mutated in a way that made it take over, yes, like as any gene with the right role could, but it would kill its host, and so
Mm.
I don't think it would leave descendants.
Okay, so it would just die out. I mean, the
Yes, I think
organism would
so.
die
I
out.
think so. Yeah.
Right. Interesting. There's a few fan-based questions at the end, but there's one from Laura Bassino, who seems to be known to you because she says we'll be in town on Friday.
Yes, former
Uh,
biology student.
yeah,
Yes.
she asks about the
Hi,
role of
Laura.
AI in the analysis of all this biological data, which is a question that would occur to a lot of us.
Yeah, so at the moment, relatively limited, although there's some potential for analyzing the large data sets that are coming out of single cell omics. Um, AI is having its primary effect at the moment in understanding protein shapes and structures and how they're interacting with each other, and also starting to in terms of understanding how genomes really work. But at the moment, we're not specifically using it in any great deal in our work.
Do you think it's a new tool you will get to use more?
I
Or is
mean,
it?
yes,
I mean, it's an evolving target. It is in history for that matter.
yeah, almost certainly. I mean, as in many any other area of science where there's reasonable quantities of data. Yeah.
Good. Nick Gillum says he's an orthopedic trauma surgeon who thanks you for a fascinating talk, but he would be interested in the development of and the variability of the skeleton across the vertebrate types, and we can understand his professional interest in that.
Yeah.
So,
Yeah.
where where does that fit into how you imagine these processes have
Well,
changed?
I would imagine that the basic processes we're talking about here would operate in other organ systems as well. So I don't see any reason why the sort of concepts of how cells, either cell type or cell complexity or organization evolve would be different between different organ systems, but it's not something we've actually studied. So I can't give you a specific answer, Nick, but I don't see why there we would expect any significant differences in the basic mechanisms of change in one tissue versus another tissue.
Right. So we can imagine that the skeleton types are evolving in similar ways across.
Yeah, and I think a lot of skeletal evolution is to do with fine scale control of size and shape as well. So
Okay.
bones are quite deeply conserved in vertebrate evolution. So
Yeah.
so we might be looking more at very subtle differences in stem cell maintenance and longevity, perhaps. Well, it's
Interesting.
not a very good answer, Nick, but it's
Well, it's a very good question, though, from an orthopedic trauma
yeah.
surgeon. You must have reflected on this in quite a lot. And the final question I'm going to read out is from the aforementioned Yuan Zhenzhu, who says, great talk. Thank you, Seb. Really learned how to give a good presentation.
I'm here, haha. I can see that bit on his
Yeah,
thing. Hi, Zen. Yeah,
well,
thanks very much for coming.
yeah. And thanks to everybody for coming today. I think that the flood of questions demonstrates just how stimulating it was for many people and demonstrates why we do these events, which is, to communicate these forms of knowledge to wider audiences. And with that in mind, I should say that the next lecture in this series is on Tuesday, the 14th of October, 5 p.m., I imagine. Raghavendra Srinivas, who's a mathematician here, will be talking about quantum adventures with trapped ions. at least have worked out how to spell ions, but beyond that, I won't attempt to explain what it'll be about. But having heard him talking on various subjects, I imagine it will be very interesting, and I hope those who are in the room this evening will join us for that. I should say in finishing that this one's been a bit special because this is the Balliol Society online lecture that we do once a year in the series that the college gives. And the whole point of the Balliol Society is, of course, that it's an organisation for all former alumni of the college. And we're always encouraging people to take part in its events. We had a very successful last weekend in Balliol. And the London Drinks, which is one of our new rituals, is going to be this year on the 12th of March. I imagine there are people in the room who've been at one of the London Drinks in recent years. And I'm told that the website now contains a link to where old members can book for the London Drinks. And perhaps they should do so because they do tend to sell out. Anyway, it only remains, I think, to thank Seb for his generosity in sharing his research with us this evening. And in the rather spooky world of silence of these lectures, I thank you collectively on behalf of everybody in the room. Thanks very much, Seb.
Thank you very much.